this is how I realized the fact that I was seeing somebody else on my PowerPoint made me realize that I had actually not made you guys each a copy. So I just made another one, and it currently says it's worth 100 points, but it's not. I just did a quick, I shoved it in there really quick. But you can open this up because there's going to be questions along the way that I want you to answer. So we're gonna watch 2 minutes and 16 seconds of a video. And then we're gonna do some science thinking and modeling, some NGSSifying of our lesson. So you have your very own copy of this. It's now not linked to the rest of the things because once I post it, I can't, like I can add and I can delete, but I can't make it available to everybody. So there's a separate one and it's not yet under the week 12. So just grab it from the top there. We're gonna watch this video of mealworms eating, and we're gonna think about some questions. We're gonna first jot down some answers of our own thoughts on this slide set so that when I put you into a group, you will have already thought about it, which would probably make for a better discussion. Okay, so just to double check, I'm sharing my screen. Yes, you see what I see? Ian Fierro? Yes, thank you. Okay, this is actually a Michigan State video.
Hey there, I'm Nina.
And I'm Daryl.
In this video series, we're going to take a closer look at the world.
Way closer.
We're going to uncover the hidden chemical changes that are responsible for the structure and functions of all living systems and support how we live. To understand them, we have to look deep inside.
From the macroscopic to the microscopic to the molecular and even the atomic scale. Today we're taking a close look at how carbon is transformed during everyday events.
Food, fuel, plants and animals, whether they're alive or dead, all contain carbon atoms and store or use energy.
We're going through an investigation and look a little closer to examine what's happening on a very, very tiny scale.
Right, and today we're feeding mealworms.
Okay, Nina, this investigation is about mealworms. We'll have a worm's eye view of these guys chowing down.
We're going to have some happy mealworms because they're gonna get served dinner. Yummy!
When an animal like a mealworm eats something, we can see on the macro scale that it chews the food up and swallows it. But what exactly is happening that's too small for our eyes to see? How are mealworms changing matter and energy when they eat, grow, and move?
What's happening at the molecular level with atoms and molecules?
We want to understand what's happening chemically when mealworms eat and grow. To do that, we ask 3 questions.
The movement question.
Where are atoms moving?
The carbon question.
What is happening to the carbon atoms?
And the energy question.
What changes in energy are taking place?
We can learn more by weighing mealworms and their food before and after they feast. BTB is bromothymol blue. BTB changes its color when carbon dioxide is added, like when you blow bubbles into it. We can use BTB to see if carbon dioxide concentrations increase in the air when the mealworms are eating. So let's make some predictions. What gains mass? What loses mass?
And don't forget carbon. What is happening to the materials with carbon atoms?
Is energy involved here? Where would it be coming from? How would it be changing? What do you think will happen? Be sure to explain your predictions as we pause the video.
Okay, so that might seem a little bit under your level, but when we want to get more at the atomic level, and I think that brings it up to the AP version. Like if I asked you originally what happens when animals eat food, you'd probably say it breaks down and absorbed into the bloodstream and then they use it to make other things, right? But we want to get deeper than that, and we want to talk at the atom level. We want to think about conservation of mass, conservation of energy, things like that, right? So we're going to bring it up to our level. So you each have, we're going to continue this another day. For now, you each have these prompts. What happens to the food that animals eat? Oh, I don't think I had that. They asked what gains mass, what loses mass. I didn't ask that, but I like that one to add. This is a reminder of the 3 questions that they asked us in the video. So the movement question, not necessarily talking about the mealworm moving, which I've never actually seen a mealworm walk. So I kind of liked that close-up view. What's happening to the atoms, and specifically carbon, which we just got done talking about photosynthesis, which was a movement of carbon. And now we're getting ready to talk about cellular respiration, more movements of carbon. So that makes for a good question there. And then what's happening to the chemical energy? Because remember, we have a couple thermodynamic laws. We have conservation of energy, and we have entropy, right? So there's a couple things we can think about there. Where are the atoms moving from and to? And then what forms of energy are in the reactants and in the products? So just take a couple minutes to quickly jot down some ideas so that when I put you into bigger groups, you can have thoughts ready for discussion, okay? So I'll give you a couple minutes to do that. Okay, I'm setting up the breakout. Okay, so I'm getting ready to throw you guys into a room. So going back to our original schedule here. So in your group, I'm going to want somebody to create a whiteboard. So I'm going to assign someone to do that. So you will go to the whiteboard, explaineverything.com, and that link is in the Google Classroom. That will give you a whiteboard that you can then invite others to collaborate on, okay? So you guys can word splash, whatnot, brainstorm some questions that come to mind. So questions maybe above and beyond the three that we gave you, right? So what would you need to know to understand this concept in a better detail maybe? Assuming you're going to run an experiment having to do with mealworms and eating, what kind of questions would you need to ask prior to running that experiment in order to be able to answer where does or what happens when the mealworms eat? What predictions you might have. So in your experiment, if you were to run an experiment, what would you expect to happen? And eventually we want to make a model. So maybe we can make a preliminary model of what's happening at the molecular level to food when animals eat it, okay? So that is what you're going to put on your whiteboard. So one person will make it. It's very easy. Sign in with your Google Classroom. And then there's a collaboration link up in the top right. And then you can share that link in your group chat. And then others will be able to collaborate on that board at the same time, okay? In the end, you will share a link to your group's collaboration in the running thread in the comments column rather than upload an assignment. Any questions over what I'm asking you to do right now? Okay, so doing some old school science department meetings stuff. Where did my breakout rooms go? Here you are. Okay, so breakout room, who's going to be creating and sharing for me? Group 1, Anna Choi, I would like you to create a whiteboard for us, please. Breakout room 2, Gabrielle Wanba. Group 3, Abigail McCursey. Group 4, how about let's let Grace Cardelio. Group 5, I'm going to go with Grace Ide. And I'm going to switch one up. I'm going to go back up to group 2, who was Gabrielle Wanba, and I'm going to ask Taylor McKenzie to lead that. Originally, I wasn't going to have the MSU people be leaders, but I think maybe I will. Okay, does anybody have to ask who's doing the whiteboard in your group? All right, so here we go. I'm going to open all the rooms. Call me in if you need me. Okay, so glycolysis, sugar splitting, basically. And there you have that. So you're going to take a 6-carbon compound like glucose, and you're going to ultimately break it into 2 pyruvates or pyruvic acid. Okay, these are 3 carbons, this is 6 carbon. Which has more energy, the 6 carbon glucose or the 3 carbon pyruvate? Yeah, there's more bonds, and if energy is stored in bonds, then the 6 carbon would have more energy and the pyruvate would have less. Is this an endergonic or an exergonic reaction? It is exergonic. Is this is it going toward or away from entropy? Toward. You're creating disorder. Okay. So this is this can be traced back to all the way to our original bacteria, right? Because they did not have oxygen. It's a very primitive process, and glycolysis does not require oxygen, nor does it require mitochondria. So you guys read about that and how it is just a single, it happens out in the cytoplasm, okay? So energy transfers first evolved in glycolysis. It's not very efficient. It's only making 2 adenosine triphosphates from every molecule of glucose. And there's much, much more energy available in the glucose molecule. So cytosol, that's referring to cytoplasm minus your organelles, right? So this is the fluid portion. Because there are no organelles in bacteria, and bacteria don't need mitochondria either. So we, however, need a lot more energy than that. So prokaryotes were the first cells. They had no organelles. They were anaerobic because we didn't have oxygen originally in our atmosphere, right? So without free oxygen, they had to have a way of creating energy that did not use oxygen, and that was simply glycolysis. It was the only step in their whole cellular respiration process. So they could take in organic molecules, break them down, release CO2. Actually, they not necessarily release CO2, but they are going to make their energy that way. So they are the ancestors of all modern life. We figure all organisms have glycolysis, which shows an evolutionary relationship between all organisms. So it must be necessary for survival. So this process is a lot like the opposite of the Calvin cycle. You have your glucose. So, you know, we ended up with glucose in the Calvin cycle. And eventually we're going to make 2 pyruvates. But in the middle, we're going to make this fructose biphosphate, which was an intermediate in our Calvin cycle. Each step uses ATP. You'll remember G3P, that was our P-gal. That was our last step before making glucose. So you can see how this is kind of similar to Calvin cycle, but in reverse. So each step requires a little bit of, it requires enzymes and then also some energy to get this started. So I need to adjust my fonts, but 2 ATP are needed to get the whole darn thing started. So even to create energy, you need to use energy, right? So there's a loss of usable energy. So we're going to use 2 ATPs. In the end, we're going to make 4. We've used up 2, so we have an overall gain of 2 ATP. Along the way, we're also going to make NADH, not to be confused with NADPH. Remember that, leave the P with photosynthesis. So just like we needed to phosphorylate PGA in order to be able to convert it into PGAL, we're going to have to phosphorylate an enzyme to make it active in order to break down P-gal. Okay, so we call this substrate-level phosphorylation. So it's using this enzyme to add inorganic phosphates. It's not using ATP. So basically, if you look at this phosphate is going to go to the edge of either of these molecules, and then you're going to strip each one off, and each one will become ATP. So if this, take 1, take 2, there's 2 of them. So you take 3, take 4, and there's where your 4 ATP come from. Okay, so we're going to keep phosphorylating in order to make that. Some sugar molecules will go through both pathways, and some will only go through one pathway. So just be a little bit familiar with that. So we need to talk about substrate-level phosphorylation. In the electron transport chain, we'll talk about oxidative phosphorylation. So the last step of glycolysis, we added those inorganic phosphates. Oh, look, PEP. Remember we talked about PEP carboxylase in our C4 plants? So we're going to use PEP again here. And by phosphorylating that, we're able to create ATP. So this enzyme will transfer that phosphate from the substrate to the enzyme. Kinase, pyruvate kinase. Any kinase you see is a protein or an enzyme that is phosphorylating another molecule. And when it does that, it either puts that molecule into an active form or an inactive form. We will see kinases in multiple situations. We'll see kinases during cellular reproduction, mitosis, and cell regulation. We'll see kinases when it comes to cell communication. So this is not a standalone concept. We're going to see that over and over again. So this is just showing you the substrate level phosphorylation, changing or moving the phosphate from the substrate to the ADP, which in this case is another substrate. And in the end, you'll end up with your ATP. So those phosphates are coming from inorganic phosphates. They're not being recycled in, like in the light reactions, we had ADP to ATP. We're not recycling them in that case. So the sugar it's using is PEP, and then it's transferring the phosphate from PEP to ADP. Okay, so it uses a kinase enzyme to do that. Then in the end, we make 4 energy molecules of ATP. So keeping track of the ATP that you're using and making, we used 2 to get it started. We made 4, so you only have a net gain of 2. At the same time, do you remember how ATP and NADPH were coupled? Like you added the phosphate and then used the hydrogen to strip it off. So NAD and ADP are also coupled, and we will be able to transfer electrons and hydrogens that way as well. Okay, so lots of similarities between this and the Calvin cycle, just opposite. Okay, so so a little bit going in and a little bit coming out, not terribly efficient. We'll talk about aerobic respiration, where we make a lot more on another day. NADH, electron carrier, just like NADPH was an electron carrier. So it's going to take these electrons into the next cycle in order to, like this is movement of atoms and molecules, right? So we're going to move it into the next step. So we take our 6-carbon sugar and we break it down into 2 3-carbons. Those 2 3-carbons are called pyruvate. You don't necessarily need to know the names of all the intermediates, but you do need to know the name of the outcome, which is pyruvate. Okay, so that's not a lot of energy, but they were little microorganisms, so they didn't necessarily need a lot of energy, right? As oxygen became available, remember our cyanobacteria started photosynthesizing, creating oxygen. So once oxygen became available, organisms started to evolve, right? And those that were better suited for the environment survived to pass on those traits. So once you had organisms that could use the oxygen, they were able to strip off more and more carbon, and every carbon-carbon bond releases energy, right? So in glycolysis, you only break 1 bond. We need to now have a way of breaking down the bonds that are in the pyruvate. Okay, so this is just showing you, look at that phosphoglycerate. We had that one in Calvin's cycle. So the raw materials, that's your food, glucose. It could be lipids. It could be proteins. We can enter the cycle in different spots, okay? So we take our glucose and we break it down into pyruvate in this case, and it's kind of fun to go through all those steps. You need to be familiar with ADP and substrate level phosphorylation. ATP, know what that means when I say substrate level phosphorylation. I'm not using ATP synthase to move phosphates, okay? NAD is going to be, is this an oxidation or a reduction?
Reduction.
Oh my goodness, brilliant people, you are right. Okay, so that is going to be a reduction. So the problem is if we don't have a way of recycling NAD, glycolysis will stop, right? So there needs to be a conversion, and that's what we're going to see happens in the next phases, is we're going to be able to recycle the NAD+. If you can't continue to glycolysize, and you won't produce very much energy. Wow, what just happened? My screen changed. So there's another molecule we can use to transfer hydrogens as well. That's going to be our FADH2. That'll come another day. So once we've made pyruvate, we can go one of two ways to recycle NAD+. That's the whole purpose. So we can either go through fermentation, which we will learn another day. That will allow us to recycle NAD so that glycolysis can continue. That's the only purpose it serves. If oxygen is available, we will follow the Krebs cycle. We have to convert pyruvate into acetyl-CoA before that can happen. So we've made glucose, we've converted into pyruvate. This all is happening out in the cytosol, the liquid portion of the cytoplasm, and then the conversion happens through the membrane of the mitochondria. So acetyl-CoA will enter the mitochondria for the next phase. And that is where we will pick up on Wednesday with aerobic respiration. Okay, so this is going to recycle NAD plus so glycolysis can continue. I'll straighten out all my letters later. So aerobic respiration, that's where the majority of our energy is going to come from. Anaerobic makes no additional energy. It just recycles NAD+. We know there's two different forms. We talked about it in 9th grade. Lactic acid, which you're well familiar with if you've ever had burning muscles, and ethanol, which makes alcohol. So depending on the organism, do you have all of the enzymes required for this very complex? If you're a yeast, perhaps not. If you're a bacteria, not, right? So you're going to go through a simpler process. Okay, so we're going to continue that on another day. Today, we just want to know what's happening during glycolysis and where it's happening, right? So cytoplasm, ultimately breaking glucose into pyruvate. We've used 2 energies, 2 ATPs. We've made 4, and we've converted 2 NADHs into NAD+. So, the stop share business, what questions do you have? Okay. I made a shortened version of a POGL. I know they can get lengthy, so I tried to eliminate things that I figured you really already know. Do I really need to ask you that? Do I need to ask you to explain this, or is the answer good enough? So I did try to break that down. And again, it didn't go in as one where you guys all had your own copies, so I re-put it in so you all have your own copies. And you're just basically going to be interpreting an overview of the first phases of cellular respiration. So that's what you got there. So it's not too long, 2 and a little bit, but they're pretty easy, so I don't think it'll take you more than 20 minutes. Wednesday, we'll pick up with our mealworms, see what's going on with them, and then we're going to continue our discussion of aerobic respiration. Okay, so get the POGL done today. I would like you to read 8.4. That's where aerobic respiration is. So I want you to do 8.4 just like you did before. Beautiful notes, summary questions at the end. That I don't need till Wednesday. So POGL today, notes for Wednesday. Questions?
Oh, by summary questions, you mean the check your progress?
Yes, thank you. Thank you for the clarification.
I got it.
Let's use different resource names and blah, blah, blah. Okay, anybody?
Miss Nook, so does that mean we're skipping over 8.3 for now?
For now, yeah, that's going to be the fermentation. So I'm going to follow through the aerobic portion and then I'll come back and hit the anaerobic.
Okay.
Yep.
For the mealworms and glycolysis in Google Classroom, if we weren't like a team leader or something, do we just mark it as done?
Yeah, that's fine. For the white,
Yeah.
that's good.
Okay.
Maybe I didn't need to put it as an assignment. Maybe I could have just put it as material. I'm learning too. Okay, good questions. Okay, keep asking or stick around for help. Check in on work you might be missing, or you guys are free to go.